Good evening, everyone. Today, we are very happy to invite Dr. Venki Ramakrishnan to give a preliminary talk in our Global Young Scientists Summit. Dr. Ramakrishnan, has determined the structure of and function of the ribosome in numerous states. In particular, his lab determined the first structure of the small subunit of the ribosome, in which we can understand how the genetic code was precisely be translated. His lab also determined the high-resolution structure of the entire ribosome, as well as a number of translational factors bound to the ribosome and a number of antibiotics bound to the ribosome. So for his great contribution and also this breakthrough, he was awarded the Nobel Prize in Chemistry in 2009. Of course, Dr. Ramakrishnan also received numerous other prizes and titles. This include, for example, he is a fellow of the Royal Society and a member of the United States National Academy of Science. And very, very importantly, he was also the president of the Royal Society from 2015 to 2020. And he also was knighted in Britain in 2012 for his great contributions to the field of molecular biology. Okay, today his plenary topic is using electron microscopy to study ribosome in action. Okay, Dr. Ramakrishnan, please.
Hello, I'm Venki Ramakrishnan. I'm sorry I can't be with you personally in Singapore due to the pandemic, but I hope you enjoy this online lecture nevertheless. So I'm now going to tell you about how the new revolution in electron microscopy has made possible our study of ribosomes in ways that we could never do before. So to begin with, here is the central dogma of molecular biology, which says that information is typically stored in DNA, which gets transcribed into messenger RNA, and that somehow gets translated into protein. And for that translation, you need this intermediary molecule called the tRNA, which has an anticodon that recognizes a codon on the messenger RNA and brings in the appropriate amino acid to add to the growing polypeptide chain. Now, of course, this doesn't happen by itself. Cell biologists a long time ago figured out that it happens in these blobs, which you can see on the left on the endoplasmic reticulum. And when those blobs were isolated, there were particles about 25 nanometers in diameter, and these particles were called ribosomes, and that's where the act of protein synthesis occurs. Now, ribosomes are enormously complicated, so even the bacterial ribosome has about 50 proteins and 3 large pieces of RNA. It's about 2/3 RNA by mass, and it consists of 2 subunits which join together to form the entire ribosome. And of course, ribosomes from higher organisms are even larger and even more complex. Now, the way the ribosome works is that the small subunit binds the messenger RNA, which carries a genetic code. The ribosome has slots for the tRNA molecules, and it somehow selects the first tRNA above the first codon, brings in the first amino acid, and then in the so-called aminoacyl slot, a new tRNA comes in, bringing in the new amino acid, and if it happens to be the correct one, the ribosome joins the first and second amino acid, and then the ribosome moves along the mRNA. At some point, the old mRNA is ejected from the ribosome, and then a third tRNA comes in, and if it's the correct one for that codon, it's also accepted, and then a bond is formed between the second and third amino acid. So you can see it's reading the code on the mRNA, choosing only those tRNAs that match the code, and then joining together the amino acids that have been brought in by those tRNAs. So it's effectively translating the genetic code into the language of proteins. Now, of course, the ribosome doesn't quite look like that simplified cartoon. Here's a picture of the ribosome where this is a sort of low resolution image from electron microscopy, and it shows that the mRNA is actually bent. It curves around the small subunit and is buried in a cleft. The tRNAs are L-shaped, and this is the tRNA holding the growing peptide chain, which emerges out of a tunnel in the large subunit. And here is the new tRNA bringing in the new amino acid. Now, of course, if you want to really understand how the ribosome works, you need a high-resolution structure so you can understand the ribosome in atomic detail, which then allows you to understand the chemistry of its interactions and thereby understand the mechanism by which it recognizes mRNA and the right codons and how the amino acids are joined together in a peptide bond. All of those things are basically chemistry happening in this very large molecule and require a high-resolution structure. Now, until a few years ago, the only way that you could get a high-resolution structure of such a large molecule as the ribosome was to use a technique called Xray crystallography. Now, in Xray crystallography, what people do is that, well, let's back up a bit and talk about how you would magnify small objects normally. So if you have a small object and you want to magnify it, you typically would use a lens, and the lens would take scattered rays from the object and recombine them into a magnified image. And then you could look at the magnified image and see details. But with atomic structures of molecules is a problem, because the distance between atoms is only about 0.1 nanometers or so, but the wavelength of light is well over 1,000 times more. It's somewhere about 400 to 700 nanometers. And so light is the wrong kind of radiation to use to look at atomic details of molecules. And the method that people had developed over 100 years ago is this method called Xray crystallography, where you take your molecule and make it form regular three-dimensional arrays. In other words, you make it form a crystal, and then you take that crystal and hit it with a beam of X-rays, and that beam of X-rays then is scattered, and in a computer, what you do is you take those scattered rays and recombine them and do effectively what a lens does. And when you do that, you can essentially generate a three-dimensional object, a three-dimensional image of the object in the computer, and you can interpret that in atomic terms. And precisely that was done for the ribosome. So in 2000, which is now astonishingly 22 years ago, there were structures of both the large and small subunit of the ribosome, and then an atomic structure of the entire ribosome was discovered a few years later. And one thing that the ribosome structures tell us is that the ribosomes probably emerged from an ancient world in which RNA did the bulk of the chemistry of life. That is, RNA not only carried information, but also did many of the chemical reactions of life, long before proteins evolved as catalysts and as structural molecules to evolve into life as we know it today. And these structures were also useful to look at antibiotics bound to the ribosome. Many decades ago, in the 40s and 50s, a number of antibiotics that were discovered were shown to work by blocking bacterial ribosomes, but not human ribosomes. And these structures allowed us to see exactly how antibiotics were bound to the ribosome. And that, of course, resulted in people trying to develop better antibiotics. Now, moving on to the more recent method, which is electron microscopy. Electron microscopy was first discovered by Ernst Ruska in 1933. He had to wait 53 years for his Nobel Prize, which was rather unfortunate. But in fact, that wasn't the longest wait. Actually, Peyton Rouse, who discovered the Rouse sarcoma virus, had to wait even longer for his Nobel Prize. So I suppose one thing, if you think you're in the running for a Nobel Prize, you should try and be very careful and not die. So how does single particle reconstruction work? Well, here's a movie made by Wolfgang Baumeister and his team at the Max Planck Institute in Martinsried that will give you some idea. So effectively, the way that this works is a small volume of a sample containing the particles of interest is deposited onto a grid, which is only about 2 millimeters or so in diameter. And then it's blotted away. So there's a very, very thin layer of liquid. And then this thin layer of your sample is rapidly frozen in liquid ethane and then studied under the electron microscope. And what you see when you look at it is you see these particles suspended in this liquid. It's not exactly ice, but you can think of it as glassy ice or vitreous ice, because it's not crystalline like ice is, but it's rapidly cooled water. And so these molecules are trapped in this vitreous ice. And when you image the molecule, you're getting projections. That is, you're getting two-dimensional projections of these molecules. And this movie shows you how you can combine all the different projections that you would see, because the particles are all trapped in different orientations. So here's what it looks. So now you have your projection of different particles. And if this is the object, then you have to relate what orientation of the object gave you a particular projection. And so you can effectively map all of the different projections onto a three-dimensional sphere, which tells you what is the particular orientation of the object that gave you that projection. And then you can then recombine those projections by a back transform to effectively recreate the object. And this is a method that was developed by many people, but initially by people like Aaron Klug and David DeRosier. And you effectively end up with a three-dimensional projection three-dimensional image of the object. Now, using exactly this method, two postdocs, Alexia Muntz and Alan Brown, and then later on a graduate student named Nirupa Desai, decided to tackle the mitochondrial ribosome. And what are these mitochondrial ribosomes? Well, mitochondria are actually organelles in our cells where a lot of energy metabolism takes place. So that is where ATP is actually synthesized. And what these organelles are very complex organelles with a double membrane, with lots and lots of enzymes in the membrane, which are part of the respiratory chain, which eventually results in ATP synthesis. Now, it turns out mitochondria actually have their own ribosomes. And the reason they need their ribosomes is mitochondria have their own genome, which encode in humans, they encode 13 protein genes. So these 13 proteins have to be made by translating the mitochondrial genome. And for that, mitochondria have their own specialized ribosomes. And when Alexei and Alan and Nirupa solved the structures, first of the yeast large subunit, and then the human large subunit, then the human mitoribosome, and finally the yeast mitoribosome, they found it was very interesting. Although these ribosomes have descended, although mitochondria have descended from bacteria, you would expect that their ribosomes look very much like bacterial ribosomes. But not only do they look very different from here's an E. coli ribosome on the right. Not only do they look very different from the bacterial ribosome, but they also look very different from each other. For example, the yeast and human mitoribosomes look very different from each other, and they're almost as different from each other as they are from the bacterial ribosome. So we could never have done this work by X-ray crystallography simply because getting enough material and being able to crystallize the mitochondrial ribosome proved almost an impossible task. And luckily, this method for cryo-electron microscopy came along and we were able to do it. So going back to the central dogma, DNA is transcribed into mRNA, and it's this mRNA which is translated by the ribosome to make protein. Now, if you look at mRNA, it's a molecule with a 5' and 3' end. And when it was first discovered, people didn't know a lot about it. They thought that maybe the entire mRNA would contain the coding sequence, that is the sequence, the codons that specified which amino acids are to be added to the protein chain. And the ribosome would simply start at one end and go off all the way to the other end and fall off. Well, it turns out that's not how it works. Actually, one of the first things that they discovered is that there's a special codon called a stop codon. Well, actually three of them, UAA, UAG, and UGA, which tells the ribosome this is the end of the coding sequence and you should stop here. And then there are special factors that come into the ribosome and cleave off the newly made protein so it can go off and do its thing. Then they realized that it doesn't start at the very beginning either. There's actually a special codon, which is always a methionine codon, an AUG codon, although it's made as formal methionine in bacteria. And that's not always at the 5' end. It is in some mRNAs, in a few mRNAs called leaderless mRNAs, but most of the time it's some way inside the mRNA. So there's a region between the 5' end and the start codon, which is called the 5' untranslated region, just as this region beyond the stop codon is called the 3' untranslated region, or UTR. Now, so then it has to know how to start at that methionine codon. How does the ribosome know where to start? So in bacteria, the ribosomal RNA in the small subunit has the 3' end of that is complementary to a short stretch on the mRNA just upstream of the AUG codon. So the ribosome knows roughly where to begin by sort of this base pairing between the ribosomal RNA and the mRNA. And then there are 3 factors, initiation factor 1, 2, and 3, along with a very special tRNA called initiator tRNA. Remember, this codes for methionine, but there are also methionines inside the protein. Lots of proteins have methionine. And those internal methionines are coded for by a different tRNA compared to the first AUG codon, for which you have a special initiator tRNA. Now, in eukaryotes and higher organisms, the mRNA is different, and in fact, initiation is much more complex. So the first thing is that beyond the stop codon, eukaryotes add a string of adenine nucleotides to the mRNA, and this is called the poly-A tail because it's a string of A's. And not only that, but the 5' end is capped by a particular diguanine residue. So it has a very special structure at the 5' end. Both of these are, to some extent, to give stability to the mRNA, so that the poly-A tail and the cap are recognized and give the mRNA some stability. But they also have many other functions, as we'll see. Now, the way initiation works in eukaryotes is, instead of the small subunit is called a 40S instead of 30S as in bacteria. The small subunit binds several initiation factors shown here, EIF1 and 1A and EIF3. And then along with that, EIF2 binds the initiator tRNA, and along with EIF5 binds this complex to form what is called the 43S pre-initiation complex. Now, at the same time, the mRNA is being bound by a factor called EIF4F, which is actually a composite of EIF4E, which actually binds that 5' cap, EIF4A, which is a helicase, that is, it's an ATPase that has the ability to unwind secondary structure in mRNA, and it has a molecule called EIF4G, which is a very large molecule, which is what interacts with this 43S pre-initiation complex. Now, the poly-A tail is also bound by a protein called PABP, called poly-A binding protein. So effectively, the mRNA can be circularized because this poly-A binding protein, there's evidence that it can interact with this EIF4G. So this complex of mRNA with the cap binding complex, the 4F complex, can recruit this small ribosomal subunit complex called the 43S complex to the mRNA, and that complex is called the 48S complex. Now, once this complex is formed, it has to scan along until it finds the start codon. That often requires ATP hydrolysis, not always. And when it finds the AUG codon, then a number of things happen. Many of these initiation factors fall off, and a new initiation factor, EIF5B, binds to this complex and recruits the large subunit. And once the large subunit is recruited, this 5B itself falls off the ribosome, and then the ribosome can go along and keep adding amino acids to the protein chain until it reaches a stop codon and dissociates, and the whole thing can start all over again. And although in eukaryotes the situation is much more complicated with almost a dozen initiation factors, there are some commonalities. So every one of the bacterial counterparts, which are known designated by IF1, IF2, and IF3 have a counterpart in the eukaryotic system. So you can think that there's a common core that's conserved between bacteria and eukaryotes. Now, there was and there still is, a kind of debate going on in the field about exactly how the small subunit is recruited to the mRNA and then how scanning takes place. So there are a couple of models. In one model, the EI4F binds to the 5' end, and it recruits the small subunit downstream of it. And this would require that the mRNA is basically slotted in to that cleft in the small subunit. And then it scans along until it finds the AUG. But this means that if the AUG is too close, for example, if it's closer than this where the tRNA would bind, and let's say it's over here, it would never be found, because this is an inaccessible region, because once it binds, it can't scan backwards because of the presence of this cap binding complex. Of course, without it, it might be able to move backwards, and there's evidence from other labs like Jody Puglisi which suggests that there might be something like that going on. The other model is called the threading model, where EI4F binds here to what's called the entry site, and then somehow the cap binding protein factor EI4E dissociates, and then this mRNA can then thread into the 40S subunit. So we decided to see what a structure would tell us. And this is all using the human system with purified factors. And it's a collaboration with Chris Fraser and Masa Sokabe at the University of California, Davis. And the people in my lab who carried out this work were led by Jay Querido, a very talented postdoc, and Sebastian Kratz, another very talented postdoc who is now doing a postdoc in computational biology in Switzerland, and Yulia Gordienko, who's a senior associate, a long-time senior associate in the lab, and Mark Scale, who did some of the mass spec experiments, which I'm not really going to talk about today. So based on the factors that Chris and Masa had purified and shown how you could assemble in vitro an initiation complex, what the lab did was to do an initiation complex and assemble it and then study it by electron microscopy. And they chose an mRNA which didn't have a start site, so that they could try and capture something that might be a scanning intermediate. Because once it reaches the start site, then these factors start to fall off. So what they found was something that looked like this. And you can see, this is the small subunit in gray, and you can see all of the very large factors that completely surround the small subunit. And you can see here are two different views of the factors of the initiation complex. So what have we learned from that? Well, the first thing is that what we've learned is that the initiation, the cap binding complex of 4E, 4A, and 4G is actually at the 5' end, and the 48S binds downstream of the cap binding complex. And what this suggests is that the mRNA was recruited by slotting the mRNA into the cleft and not by threading. So it's not like the cap binding complex is binding here at the entry site and then somehow the mRNA is being threaded. Rather, it's binding at the 5' end and recruiting the 40S downstream of it. Now, when it binds, it must somehow pull the mRNA through it. And there's some debate about whether the cap is released or whether the foray actually hangs on to the cap, in which case if the mRNA is pulled through, it must sort of loop out like this so that it's still bound to the cap binding complex. So at least the work we've done so far very strongly supports the idea that the cap binding complex actually recruits the 43S complex and the mRNA is slotted into the 43S complex, and some sort of pulling mechanism then pulls the mRNA through it. Now, if you now look at the entry site, in this structure, the entry site was completely empty. There didn't seem to be any factor bound at the empty site. But Jay Querido has now done a structure as part of the same collaboration with an mRNA that has quite a bit of secondary structure, and he's used a trick to lock foray, which is an ATPase, so that it won't fall off and it'll be locked in place. And when he does that, he not only sees the 4F, which also includes a foray, which is the helicase, at the exit site near the 5' end, but he sees a second foray bound near the entry site of the mRNA. So this suggests that you might have a second foray, and you might need a second foray in many cases. For example, if you have highly, if you have an mRNA with a lot of secondary structure that has to be unwound, then you might need a second foray ahead of the 43S to unwind the mRNA as it scans. And so here's a model for how scanning might work. And that is, you have your cap binding complex with one of the forays at the exit site. It has a certain role. And then you also have a second foray, or it could be some other helicase. For example, there are helicases like DHX29, which may also serve that function. And that binds near the entry site and may be used, for example, to melt or unwind secondary structure in the mRNA in order to reach the start codon. So I just want to summarize by saying that cryo-EM has made possible the determination of structures that were never before possible because of the amounts of material you could get, or because they were unstable and so on. The reason it's so powerful is that you need very small amounts. You need a thousand-fold or less material compared to a typical crystallographic project. And the samples don't need to be, this should be homogeneous. They don't need to be homogeneous. They can be heterogeneous. And you can capture transient or dynamic complexes. And if you use tomography using cryo-electron microscopy, this is a technique which allows you to look at entire cells or sometimes slices of cells. So you can look at complexes as they exist in situ, inside cells. So all of these revolutionary advances means that we're really in a new era for structural biology where amazingly difficult projects can be tackled and important new discoveries can be made. Thank you very much.
Okay. Dr. Venki Ramakrishnan, thank you so much for a very, very wonderful talk. Yeah, it is crystal clear. Yeah, I always enjoy your your talk, your presentation. Yeah, so maybe we will have a question from the audience, but let me begin first. So as for the mitochondrial ribosome, and also you compare the mitochondrial ribosome with the like a loma, like a bacterial ribosome, etc., then you found the mitochondrial ribosome more like a diverse. So you also mentioned like you early discovery, like a number of antibiotics bound to the bacterial ribosome, and then this can rationalize how the molecular mechanism of the antibiotics, and also is very, very useful for the new drug discovery. So my question is that, do you think the mitochondrial ribosome would be a good target for like a drug discovery to tackle the new like some antibiotics resistance crisis?
I think because we all have mitochondria, if you target mitochondrial ribosomes, it will be toxic to us. And in fact, it's believed that one of the reasons that many ribosomal antibiotics are toxic, for example, chloramphenicol or some of the streptomycin and so on, have side effects, is because they bind to our mitochondrial ribosomes, because they're closer to bacterial ribosomes, at least the antibiotic binding site. So there is an idea, though, that you can target mitochondria for cancer therapy, because cancer therapy, cancer cells use a lot of energy, and since mitochondria are the source of energy in the cell, if you target mitochondria, it might preferentially affect cancer cells, but it hasn't really. I don't think it has worked so well. Okay, so I think the more important thing is it will allow you to design antibiotics that bind to bacteria but do not bind to mitochondrial ribosomes. And that way it'll be more specific and have fewer side effects.
Okay, I see. Thank you. Okay, so let me check from the question from the audience. Okay, so we have a number of questions from the audience. So like one from John Benita. Her question is there a typical 5' UTR consensus sequence like the needed for proper initiation slotting, or is it a generic non-specific interaction with the ribosome, or no need?
Yeah, I think there are two things here. One is the initial recruitment of the mRNA, and the second is efficient initiation at the AUG. So I believe that for the initial recruitment, it's relatively non-specific. It's for any single-stranded mRNA. Of course, it has to be in the proper context with, cap binding complex, etc. But when you get to near the start codon, I think that's where the Kozak sequence will make it more efficient. And same with Shine-Lugano in the case of bacteria.
Okay, thank you. So we have another question from Shu Bao Zhao. Fantastic talk. So the question is that how do you think about the appearance of AlphaFold, where it influenced the research of structural biology, and what will be the research emphasis in the future of structural biologists?
I think AlphaFold is I think it's a very big breakthrough because it does allow you to calculate what the fold of a novel sequence might be. It is very early days, and so we do have to get some idea of where AlphaFold works and what fraction of the predictions are accurate and what fractions have problems. But I imagine that in the next few years, machine learning will really make a lot of progress towards fold prediction. But then we have to think about how proteins change conformation, how they bind ligands, how they interact with each other, and so on. And it's very hard to predict, but my guess is that machine learning will really help structural biologists a lot. I don't think an empirical experimental structural biology will ever go away, because we need that to, first of all, to be sure, secondly, to see subtle features and so on. But I think it'll be a great tool. It'll be a very, very important tool in structural biology.
Okay, great. Oh, thanks for a great lecture. What do you think are the new directions in ribosome research? Maybe she mentioned, oh, maybe example time-resolved EM, or how can it be applied in medicine?
Yeah, I think there are a number of questions. I think a lot of the focus in the last 20 years has been on understanding the basic structure and some of the basic mechanisms in what you would call canonical translation. And I think the future will be more directed towards control of translation. How does the cell regulate translation? Because that's a very, very important aspect of gene expression and biology. Another thing is about non-canonical translation. For example, viruses can avoid many of the normal steps during initiation of translation, and they have their own mechanisms. How do RNA modifications affect initiation and translation? So there are a lot of questions which have to do with regulation and altering the normal course of translation and quality control and so on.
Okay, thank you for the clear answer. Okay, then the one question is that thank you for the excellent talk. I have one question about the evolution of translation. Are there any transitional steps in evolution between bacteria initiation and the eukaryotic scanning model?
Yes, that's a very interesting question. Clearly, the core is conserved because the three initiation factors in bacteria have counterparts in the eukaryotic initiation factors. So there is some common conserved core to this complicated process. I don't know if we have real intermediates. I suppose one could study archaea, which eukaryotes have sort of evolved from archaea. So one could study archaea and initiation of translation in archaea. And perhaps that's a somewhat understudied field, and maybe that's something that deserves more attention.
Thank you for a great talk. Even though CryoEM is powerful, what are big limitations of this technique?
That's very interesting. So one of the limitations, of course, is as you get down to lower molecular weight, say below 40 kilodaltons or 50 kilodaltons, it becomes progressively harder or impossible to get enough signal to align the particles and obtain a structure. So that's something that people are working on, is how to improve the signal to noise in cryo-EM. Another problem is that often the core of the molecule will be very stable and well determined, but in the periphery where you are binding, factors and so on, there's a lot of dynamics. Those periphery are not fixed relative to the core, and so as you go out from the core, the resolution becomes worse. And so that is another real problem with cryo-EM. And finally, some complexes are highly dynamic. They have an entire ensemble of structures, making it hard to, determine any one of them to high resolution. I think, though, experimentally, these problems are being tackled both by improving the actual microscopy, by better sample preparation and grid preparation, and also by software, by improved algorithms. So CryoEM is not a static field. It's still early days. It's a bit like crystallography was in the 1970s, and people are making dramatic improvements all the time. And of course, as we discussed before, there are entirely computational techniques like AlphaFold, which will only help CryoEM. You know, if you have a map that's not quite interpretable, but then you have an AlphaFold model, and you can constrain AlphaFold with the model, that'll actually be more powerful. So I sense that there are going to be big improvements in structural biology.
Okay. Yeah, thank you. So there are still many questions from our audience. Yeah, because I think you gave a very wonderful talk, very clear talk, so they understand, and then they have more questions. But there's a time limit, so unfortunately we cannot continue our Q&A session. So once again, with a big thanks to Dr. Venki Ramakrishnan. Thank you very much for a wonderful talk. Okay, see you.
Thank you.
Hopefully
I'm
one
sorry.
day you come to visit Singapore.
Yes, I'm sorry I couldn't come this year. And let's hope that in future years things are better and I can visit Singapore again. Thank you.
Yeah. Warmly welcome. Okay, thank you.